and pleasant afternoon to all. It is my great pleasure to welcome you to the opening of the OMICSena lecture series. Some of you might have attended first OMICSena last March. That was an information field premiere of the OMICSena platform where DOST-FNI featured the frequently asked questions on nutrigenomics. For the second time, DOST-FNI brings you OMICSena with a bolder move to bring together omics experts from way beyond the Philippine borders. The Philippines continues to build strong innovative foundations, and learning and reflecting from the global scene is a very way to go. As such, we are thrilled to bring you the 9-day OMICSena lecture series. For 9 afternoons, we are expecting to learn the best of omics technologies and how these emerging platforms can enhance nutrition and food research that will eventually be translated for societal benefits of our country. Beyond basics, hopes are high that we can also build networks and collaborative paths, all towards building an omic-driven, innovative Philippines. William Wordsworth said that life is divided into three terms: that which was, which is, and which will be. So let us learn from the past to profit by the present, and from the present to live better in the future. I hope that through all the topics to be discussed from this day until October 7, we can learn how to create a better future in life and nutrition through the studies of omic sciences. To all the participants, again, my warmest welcome, and lend us your active participation because we have inspiring lecture series ahead. So thank you so much and stay tuned. Maraming salamat po at magandang araw muli.
Magandang agham po sa ating lahat. OMICSena was coined by DOST-FNRI to create a scene in a good way. It seems that it will continue to serve as a platform for the DOST-FNRI to keep us in the loop on its undertaking in genomics. With Dr. Brian Gonzalez on board, the second OMICSena for 2021 collaborates with the DOST's Balik Scientist program to drumbeat the advent of another set of omic science at the Institute, the metabolomics. This expansion in the R&D platform will guide DOST-FNRI in setting up more actionable strategies in the molecular food and nutrition arena. I am optimistic that the series will provide new learnings to bring stronger and bolder omics-based research and development initiatives, not only in DOST-FNRI, but in the entire country, as I understand that this OMICSena is well represented by genomics enthusiasts mula Luzon, Visayas, at Mindanao. As we come together for the next 9 afternoons, I hope that we will be able to lay out plans and strategies towards the creation of new intervention systems, biomarker discovery, and innovative health and nutrition platforms through the use of omics technologies. May this event sharpen our vision to acquire the lens of seeing through what our country needs to reach zero hunger and optimum health and nutrition at the molecular level. Congratulations to the organizing committee of the OMICSena lecture series and to all the staff of DOST-FNRI led by Dr. Imelda Angeles Agdepa. Magandang agham sa ating lahat.
A pleasant day to all. The Nutritional Genomics Program of the DOST-Food and Nutrition Research Institute is part of the DOST's Big 21 in 2021. This achievement highlights the growing field of nutritional genomics and the DOST-FNRI's New Gen Laboratory, now an ISO 17025 Certified Molecular Laboratory. Beyond this recognition, I am more thrilled to see what our R&D program and scientific and technological services in nutritional genomics can offer to ensure optimum nutrition for everyone. Earlier this year, the first OMICSena answered frequently asked questions on nutrigenomics. The first installment of this web-based learning platform of DOST-FNRI's genomics programs made us realize that nutritional genomics can scale up nutrition and health sciences. Studying our genes through the omics platforms can bring about enhanced nutrition recommendations and perhaps nutrition and health policies in the long run. Its eventual integration in clinical practice can boost our efforts to overcome malnutrition and help us achieve our goals of good health and well-being. As DOST continues to expand the country's omics footprint, this second offering of OMICSena with Dr. Brian Gonzalez, our balik scientist for metabolomics, is anticipated to bring new frontiers in omics-based innovations. This will fine-tune our efforts to use various molecular platforms to pacify and eventually end our long-standing battle against malnutrition and non-communicable diseases. Now, with the generous sharing of information and practices from our keynote speakers ahead of us, I invite you all to join me in getting a detailed glance at omics technologies and how they can play critical roles in nutrition and health. May this deep appreciation of omics technologies guide us in pushing for more innovations directed to our local needs. I am optimistic that this lecture series will also strengthen the competencies of our researchers in initiating omics-based research and development. One thing that the ongoing pandemic has taught us is to hold tight to what science and technology can do. Science and technology will always be an important key in solving various societal problems. Napaghahandaan man o hindi, katulad nitong pandemya, innovations and discoveries will help us thrive and become resilient. On behalf of the Department of Science and Technology, I would like to thank our speakers from different parts of the European Union and the United Kingdom for their time and expertise. I extend my appreciation especially to our DOST Balik scientist, Dr. Gerard Brian L. Gonzalez, for bringing this exciting series to light, together with the DOST Food and Nutrition Research Institute. Thank you all for being here. Let us have a learning-filled week ahead. Mabuhay!
In 2010, DOST-FNRI revolutionized its take on nutrition by incorporating genomics in its researches, investigating the relationship of anthropometric, biochemical, clinical, dietary, and health components to the genetic makeup of Filipinos. DOST-FNRI is looking into providing a more personalized approach of achieving optimum nutrition to every Filipino. Through nutritional genomics, we attempt to better understand how a single nucleotide, a single letter, can make a big difference in our way of living. And so, to support this endeavor, DOST-FNRI capitalized on establishing its molecular laboratory known as the Nutritional Genomics or the NewGen Lab. With its thrust of bringing science for the people, The new Gen Lab provides assessment of genes and genetic variants associated with micronutrient deficiencies and diet-related non-communicable diseases. The new Gen Lab is equipped with some of the state-of-the-art equipment in molecular biology, such as the automated nucleic acid extraction machine, bioinformatics, real-time and digital PCR systems, automated liquid handling system, micro-volume spectrophotometer system, and gel documentation system. With a complete workflow adhering to the highest standard of competency in laboratory testing, New Gen Lab is setting the mark in providing mole biotechniques with transparency, validity, and reliability. Whether you are an individual or a group of enthusiasts seeking answers in nutritional genomics, the New Gen Lab awaits you. Our proficient personnel works in close contact with our customers through customized analysis, provision of technical consultancy in nutritional genomics, and standardization or laboratory competency. To even serve you better, we are partnering with a leader in genomic testing for a higher throughput and faster turnaround time. Welcome to the new generation of nutrition. Welcome to the New Gen Lab.
The FNRI Service Laboratory is an ISO IEC 17025 accredited laboratory which is composed of 3 laboratories. It is the focal agency of the Department of Science and Technology for Nutrition Labeling and is recognized by the Philippine FDA for nutrition labeling and by BFAR for marine food products analysis. It is the only laboratory in the Philippines that is capable to analyze serum vitamin A and urinary iodine excretion. Initially mandated to conduct researches on biological parameters and support the laboratory needs of various in-house food research and development projects, FNRI-SL has continuously expanded and has offered its services also to the public. The chemical laboratory is equipped with instruments that allow proximate determination and physical-chemical analysis of foods. Also, our laboratory is equipped with state-of-the-art modern instruments such as high-performance liquid chromatography, gas chromatography, atomic absorption spectrophotometer, graphite furnace equipped AAS, automatic analyzer, WYD iodine checker, and the UV-Vis spectrophotometer is currently used for developing methods and water quality assessment. In the biochemical laboratory, other modern instrumentation are being used in order to support the National Nutrition Survey, medical research institutions, LGUs, non-government and international organizations. Having continuously participated and showed satisfactory results in the quality assurance programs of the U.S. Centers for Disease Control and Prevention, the Biochem Lab offers now a wide range of tests. Meanwhile, the microbiological lab is a food laboratory offering range of tests covering spoilage, indicator and pathogenic microorganisms in food and water samples. The microlab offers the following: Also, the microlab conducts isolation and identification of pathogenic microorganisms in food that are of significance to public health. Recently, the laboratory has also started performing molecular pathogen detection in food and bacterial cultures. Using the technology of real-time polymerase chain reaction, detection of Salmonella and Vibrio has become more rapid and accurate, as we are committed to deliver high-quality and timely services. The laboratory is maintained by well-trained, experienced, and PRC-registered chemists and chemical technicians, medical technologists, and registered microbiologists on analysis of different samples. Aside from our commitment to maintain quality data generation, FNRI SL also holds trainings and continuous consultation to customers and stakeholders. In the future, the laboratory aims to expand its services by offering specialized tests to the public. In order to do this, we commit to continuously develop methods and train our laboratory staff for excellence and quality service.
Day 9 of 9. Magandang hapon Pilipinas and good morning Central Europe. OMICSena lecture series is grateful to the inspiration given to us by the Honorable DOST Secretary Fortunato de la Peña, DOST Undersecretary for Research and Development, Dr. Rowena Cristina L. Guevara, and our dear DOST FNRI Director, Dr. Imelda Angeles Agdepa. Joining us this afternoon is our Deputy Director, Dr. Anthony Calibo. Good afternoon po, sir. We are down to the last episode of this 9-day run of the OMICSena lecture series. This early, I would like to thank everyone for joining us every single day for the last 8 days. We hope we were able to meet your expectations. As we're about to pull the curtain to close the second season of the OMICSena lecture series, we will have another exciting and learning-filled discussion this afternoon. Before Dr. Brian Gonzalez introduced to us our expert for the last time, please allow me to inform everyone that the OMICSena lecture series is part of the countdown to FNRI's 75th anniversary. The Institute will be staging more events to drumbeat our anniversary on July 1, 2022. Ang DOST FNRI ay kasama nyo sa bawat yugto ng buhay at kabuhayan for 75 years and beyond. An important force behind the OMICSena Season 2 is our DOST Balik Scientist for Metabolomics, Dr. Gerard Brian Gonzalez, who will introduce to us our speaker this afternoon. Over to you, Dr. Brian. Good afternoon.
Maraming salamat and magandang hapon ho sa inyong lahat and good morning to our guests here in Europe. Maraming salamat ho sa inyong patuloy na pagsubaybay sa ating OMICSena lecture series. Ngayon ho ay ang ating huling araw ng webinar at kinalulugod ko hong ipakilala sa inyo ang ating bisita ngayong araw, Dr. Liselot Hemerick from Ghent University in Belgium. Dr. Hemerick is currently a postdoc and a research and teaching assistant at the Lab of Chemical Analysis, led by Professor Lynn van Hacke, whom you have heard yesterday, at the Ghent University in Belgium. She started her career in 2012, focusing on the development of a DNA adaptomics platform to study the genotoxic effects of both endo and exogenous chemicals. At the time, Dr. Hemmerich pioneered the study of DNA adaptomics, being one of the only 4 developed holistic high-resolution DNA adaptomics methodologies worldwide. And for that, she has received prestigious awards in Belgium, including the Prijs van de Academie voor Wetenschappelijk Onderzoek in de Diergeneeskunde 2019, which in English is the prize from the Academy of Scientific Research in the Veterinary Sciences, which, by the way, Ghent University Veterinary Sciences Department is consistently #1 in the world for veterinary medicine. And then we're very proud of that. I'm a proud alumni of Ghent University, of course. And her research interests involve environmental and food toxicology, mass spectrometry-based metabolomics, as well as further development and optimization of mass spec-based DNA adaptomics methodologies and data analysis workflows. So without further ado, mga kababayan, salubungin ho natin ang masibabong palakpakan, Dr. Liselott Hemmerich. Liselott, the platform is yours.
Yes, thank you. I will share my screen. Okay. Which should be okay, I think.
Yes. We can now see your screen, Liselott.
Yeah, thank you for confirming. So good morning, everyone, and I think to most of you, good afternoon. Indeed, as Brian mentioned, my name is Lies Lotz, and I'm currently a postdoc at Ghent University. And today, I'm very grateful that I will be able to talk to you about mass spectrometry-based analysis of the DNA adductome, also called DNA adaptomics. So, as mentioned, I'm based at the Laboratory of Chemical Analysis at the Faculty of Veterinary Medicine in Ghent University, Belgium. And besides teaching, we are also involved in a lot of research, and this actually takes up the vast majority of our time. And we are mainly focused on the analysis of small molecules using mass spectrometry. Now, as Professor van Hacke explained yesterday, and some of you may have followed her lecture yesterday, a large amount of our work focuses on, excuse me, on metabolomics and lipidomics in health and disease. But besides metabolomics and lipidomics, we also have a research line which focuses on DNA adductomics, which started out with my PhD a couple of years ago. Now, an overview of what I would like to present to you in the next 45 minutes or so is the following. So first of all, I would like to introduce you to the topic of DNA adaptomics and discuss the technology as well as the methodology, the workflow that is required for the analysis, and then also some examples from our own research to demonstrate actually the potential of this method. And then also further optimization and requirements for methodology as well as data processing workflow, followed and closed by some take-home messages. Now, I am aware that DNA adductomics is less well known than metabolomics. So for those of you who are not familiar with the concept or the term, I think it is best for me to start my talk today with explaining to you what DNA adductomics is exactly. So DNA adductomics, somewhat in line, of course, with the other omics technologies and methodologies, is the study of all DNA adduct types and levels within an organism, organ, tissue, or cell. And DNA adducts itself are actually compounds that are formed upon the interaction of an electrophilic reactive molecule with DNA. And this can happen at different reactive sites within DNA, being at the nucleobases, so adenine, guanine, thymine, and cytosine, but also at the sugar moiety and the phosphate group. Now, all in all, most commonly adducts are formed at the N7 and N3 position of guanine and adenine, but depending on the chemical and the interactions, there are a lot of different DNA adducts that can be formed by one and the same chemical. Now, regarding those endogenous and exogenous chemicals that can induce DNA adduct formation, it's important to note that there is a very wide range of chemicals that are indeed able to form DNA adducts. And this can grow from chemicals that originate from outside of our body. So, for example, including man-made chemicals, but also naturally occurring chemicals, including, for example, mycotoxins, as well as chemicals that are formed within our body under normal, healthy circumstances, but also pathophysiological circumstances. So, for example, within our body and during normal homeostatic metabolism, we, for example, have the formation of reactive oxygen species, and those reactive oxygen species can induce DNA adduct formation. It's a normal process. But of course, depending on what we're exposed to throughout our lives, these processes can be induced, can be stimulated, and there can be more DNA adduct formation than is actually required or foreseen. Now, besides the natural physiological processes in our body, we can also, like I said previously, be exposed to chemicals from outside. And these chemicals are also metabolized within our bodies and can then, as a metabolite, induce DNA adduct formation. And then a final example, which I will come back to later on during my presentation, is that also the microbes in our guts can induce DNA adduct formation by producing certain metabolite products from our food. Now, when DNA adducts are formed and when they are left unrepaired and accumulate over time, these DNA adducts can induce mutations and can thus induce pro-carcinogenic effects. And two established examples of this is, for example, aflatoxin B1, of which it is known that this compound is metabolized in the body, forms DNA adducts, and specifically DNA adduct formation is linked to carcinomas, as well as, for example, aristolochic acid, which also induces cancers in the urinary tract. Now, these are well-known effects of DNA adducts, although the field is most definitely still understudied and there is still a lot to research. Lately, it has become clear that DNA adducts may also interfere with normal cell function and that they may interfere with transcription, replication, and thus may or may not, this is still under debate and should be investigated, may lead to developmental aberrations early on in life, so in newborns and in young children. So in environmental monitoring, we can use DNA adaptomics to map all DNA adduct types and levels. And this actually allows more in-depth investigation of environmentally induced DNA damage. Because DNA adducts actually give us information on not only exposure to certain toxins or genotoxic chemicals, but also on their metabolism. Of course, whether or not they form DNA adducts, whether or not those DNA adducts are repaired, and then also last but not least, whether or not those adducts have certain adverse health effects. So can they induce cancer? Do they increase our cancer risk? And because of this, DNA addicts or DNA adduct analysis has two specific advantages. One being that they are stable markers of exposure and the biologically effective dose on the one hand, and also that there are markers of effect. So effect in the body, DNA adduct formation is an intermediate effect, but as I mentioned previously, it could also be a marker of increased cancer risk. So continuing on the example of aflatoxin B1, I would like to demonstrate that with this. So aflatoxin B1 is mycotoxin. And if we are exposed to aflatoxin B1 and ingest it, it can be metabolized to its epoxide form. This epoxide formed can then lead to the formation of DNA addicts. So when detecting these addicts in the body, we know that we have been exposed to the mycotoxin, and we know that it has been metabolized. If the DNA addict is present, it also means that this addict has been accumulating over time. So we have been exposed chronically. And as we know that the DNA addict is linked to an increased risk of carcinoma of the liver, we know that this addict is actually a marker of increased risk. So you have it on both sides. It marks exposure and it marks potential effect. Now, when wanting to study these DNA addicts and wanting to analyze them, we of course need a fit-for-purpose methodology. And it's important to note that over the years, several methods have been developed for DNA addict analysis, but more specifically focusing on the targeted analysis of DNA addicts. And one of the first methods that was used and is still being used was P-post labeling. But also immunoassays, and then later on, a lot of mass spectrometry methods were developed. Now, in targeted analysis and using these three methods, we can analyze a limited number of known DNA addicts, like 1, 2, 3, maybe up to 10. But in any case, we need to know what we're looking for beforehand. In untargeted analysis, in line with metabolomics, we also want to look into unknowns. So we do not only want to look into the occurrence of DNA addicts that we know and are important, but we also want to look for those that we do not know yet, but may be very important as well. For untargeted analysis, we cannot revert back to P-post labeling or immunoassays because they're solely targeted, but we need to go into high-resolution mass spectrometry. That is, in fact, the only option for untargeted DNA addict analysis to allow the analysis of a wide range of DNA addicts for screening of both known and unknown DNA addict types. Now, going back to the targeted DNA addict analysis, I would like to point out the specific advantages and disadvantages of the different techniques, because, of course, this matters. For P-post labeling, two very important advantages is that they acquire a very low amount of DNA. And since the amount of DNA is always a problem within DNA adaptomics, this is a really big advantage. And it's also a really sensitive technique. Now, the disadvantages, on the other hand, are that you have no structural information about the DNA addicts. So you cannot learn more about a new addict or learn more about its structure. And the technique is also not fit for quantitation. Another downside is that there are a lot of false positives and that it's also labor intensive. Regarding the immunoassays, as opposed to the P-post labeling, we have the opportunity to perform semi-quantitative analysis. And also it is possible to visualize the actual position of the addict in tissues or cells. Again, this technique does not allow for obtaining structural information. There is no absolute quantitation, and there are also a lot of false positives due to cross reactions. Coming to mass spectrometry, there are a lot of advantages with the method being both highly specific and highly sensitive, furthermore allowing absolute quantitation and also providing structural information of the DNA addicts on the research. Major downsides, or I think you are all well aware of this, is that's a rather expensive technique and that it requires skilled personnel to handle the methods and technology. So as I mentioned, for untargeted DNA addict analysis, mass spectrometry is the only option, and then specifically high resolution mass spectrometry, because we need high-end analytical instrumentation to enable accurate mass measurements and screening of, well, a virtually unlimited number of compounds. So DNA adduct analysis by mass spectrometry can be optimized using a high-resolution instrument. And it is important to mention that there are different ways in optimizing your method and using it, depending on the form of the adduct you want to measure it in. Now, just to clarify, because that's important throughout the rest of the presentation, you can measure adducts in their nucleotide form, in their nucleoside form, or their nucleobase form. And whether or not you want to do it in one versus the other, this requires specific settings during mass measurement, as well as sample preparation. So, as noted here on the slide, when measuring the base during sample preparation, you will need to cut off all the rest. You only want the adducted base, which is demonstrated with the green arrow. When you want to measure the adduct in nucleoside form, you want to set up a procedure that will cut out the DNA adducts with the base and the sugar. And when wanting to measure it in its nucleotide form, you need to cut it out apart from the DNA, but with the entire entity of the nucleotide. Now, the most commonly used approach is the nucleoside method. And following up on that, it's the nucleobases. And whether or not you decide to do one versus the other, you need specific settings. Now, a couple of years ago, during my PhD, I developed an ultra-high performance liquid chromatography coupled to high-resolution mass spectrometry DNA adaptomics method, which allowed both targeted and untargeted assessment of DNA adducts using a hybrid instrument, which was in fact an quadruple Orbitrap instrument. So this hybrid instrument had the specific advantage that it was both highly sensitive as well as specific, allowing the identification of unknowns with high certainty and the quantifications of relatively low levels of DNA adducts. The method was developed and optimized and also validated according to FDA guidelines, but I won't bother you with the details on that. More specifically, what could the method do, or what was envisioned with this method was the targeted detection of 4 DNA adducts of interest, being 0,6-methylguanine, 0,6-carboxymethylguanine, the major malondialdehyde adduct with guanine, and the major crotonaldehyde adduct with guanine. So these are in fact alkylation and lipid peroxidation adducts, and these are of importance to our research project specifically because they may be related to red meat intake. I will come back to that later on. But coupled to the targeted detection, we also envisioned untargeted detections because besides these 4 adducts, we know that there are many, many more other adducts that may be of relevance in diet-related carcinogenesis. So we wanted a really broad method that allowed for full scan analysis and allowed for screening and possible discovery of new adducts. And to that purpose, we also coupled this to an in-house DNA adduct database, which at the time had about, I think, 180 DNA adduct names and structures in there, and has now been expanded to over 300. So based on literature to facilitate the identification of unknowns. Now, besides the method itself, DNA Adaptomics also requires its own workflow, including sample preparation and also data processing. The way we perform DNA Adaptomics is very much in line with metabolomics. So for those of you who have attended the previous lecture, some parts will be very familiar and it will be easy to step into this workflow. But of course, DNA adducts are not metabolites. So there are certain steps that are different and need to be optimized for this method specifically. Now, going through the workflow, the very first step is, of course, always sample preparation. And in DNA Adaptomics, the first thing you need is, of course, DNA. So you need to extract your DNA from your samples, from your studies. After extracting DNA, the next step is obtaining those nucleotides or nucleosides or nucleobases, and that you need to perform that you need to obtain by performing hydrolysis. And depending on the way you want to go, there are two options, being enzymatic hydrolysis or thermal acidic hydrolysis, to be able to obtain the nucleotides and nucleosides with enzymatic, or the nucleobases with thermal acidic hydrolysis. Now, after obtaining the individual nucleobases or nucleosides or adducted bases and nucleosides, the next step is that we want to do an extra cleanup and concentration of those samples to improve sensitivity. So overall, DNA adducts levels are relatively low in DNA. There is at the time no possibility to multiply them. So the levels are low and we need to enrich the sample to be able to measure them with sufficient sensitivity. So sample cleanup is, for example, most often solid phase extraction using small columns. Now for the analysis, so following up on the sample preparation, we get to the actual analysis, where in parallel we can perform the targeted analysis using MSMS, and on the other hand, then the DNA adaptomics analysis using high resolution mass spectrometry in full scan mode. After the analysis comes the data processing. And the data processing in DNA adaptomics as well as metabolomics can go from a simple t-test to just compare averages in one group versus the other, or into multivariate statistics with PCA modeling and OPLSDA modeling to look into the markers. So regarding the targeted data, this is quite straightforward and in line with other mass spectrometry-based analysis. So for the identification, we use analytical standards, and for quantification, we work with a calibration curve. For processing of the data, we use dedicated software. So depending on the system you're using, this will be a vendor-specific software or maybe even in-house software you developed yourself. And then statistical interpretation is done using univariate statistics, t-tests, ANOVAs, etc. So this is quite straightforward and will be easily Then regarding the untargeted data processing, this is in line with metabolomics. We have, first of all, pre-processing of the data, and secondly, processing. The pre-processing allows for peak picking and alignment, which can be performed with commercially available software like Compound Discover, or in-house software like we are also developing ourselves, followed by normalization, transformation, and scaling of the data in preparation of the actual processing. And like I mentioned, in the processing, we use multivariate statistics to describe the difference between two groups of interest by means of, for example, PCA analysis or OPLSDA analysis. Again, this can be achieved using vendor-specific software commercially available, like, for example, Simca, or also in-house software. Now, after looking into the workflow, I think it will be of interest to you to see what actually happens with the workflow and the data and what we can do with this technology and methodology. And therefore, I have prepared some slides of the work we have done previously in the context of our research on red meat-related carcinogenicity in relation to colorectal cancer specifically. So to be able to go through these results, I will first give you a small introduction on the topic. And you may or you may not be aware, but when you eat too much red meat, you increase your risk of developing colorectal cancer. So in fact, when you eat 100 grams of red meat per day, you increase your risk with 29%. Now, the main reason behind this is to date still unknown. We don't know what the exact underlying mechanism is, But the main hypothesis on the difference between red meat versus white meat, so red meat is pro-carcinogenic and white meat is not. And the main hypothesis on this is the heme hypothesis. So in red meat, there is more myoglobin present than in white meat. And myoglobin, which is a protein, contains heme iron. And the hypothesis is that heme iron might be pro-carcinogenic, although we do not know exactly why that is. So heme iron may exert direct and indirect effects within our bodies when it is digested. The direct effects, what we know is that heme iron can be cytotoxic, although there may be other effects that we are unaware of today. But it can also be indirectly toxic by inducing the formation or metabolization of certain compounds. So heme iron can induce the formation of harmful N-nitroso compounds and also lipid peroxidation products. And those N-nitroso compounds and lipid peroxidation products can be both genotoxic and cytotoxic. And in this way, heme iron may, in the end, initiate, promote, or aid in the progression of cancer. So in this context, we wondered whether or not red meat versus white meat, and then specifically also heme iron under the form of myoglobin, induced the formation of specific types of DNA adducts. So is the DNA adductome significantly different when consuming red meat in comparison to white meat? And we set up different studies to investigate, in which we used our DNA adaptomics methodology to map the DNA adaptome of red versus white meat digests in vitro, and also in tissues from rats who were fed red versus white meat. So this was done by means of two models, and just for clarification, I will explain what they are exactly. So the first model we used was an in vitro digestion model, in which we simulate the digestion of a certain food or feedstuff. So what we do is we actually grind the food or feed, add stomach juices, add intestinal juices, and also add the colonic microbiota of healthy donors. So this is a very flexible system in which we can add different juices or bacteria at different time points, and we can also sample at different time points. So chronologically, we simulate the stomach, small intestine, and large intestine in this setup. And then following up on this, we can take samples, extract the DNA adducts, and analyze the DNA adductome. The second model was an in vivo rat model, which were in fact Sprague Dawley rats, which were fed a red or white meat-based diet for 14 days. And after these 14 days, the rats were euthanized, and liver, small and large bowel tissue were sampled and prepared for analysis. Now, we performed several independent experiments. In total, there were 4, but today I will briefly show you the results of 3 of them. So the first 2 were in vitro digestion experiments, in which we wanted to see whether or not there was DNA adduct formation during digestion, and whether or not there was a difference in red versus white meat, and also investigating the effects of calcium and myoglobin. So myoglobin, I already explained. The hypothesis is that when adding myoglobin, there would be more DNA adducts. With calcium, it's the other way around, because calcium is believed to have protective effects. So calcium was experimented with to see whether or not this influenced DNA adduct formation and made us be protective. The third experiment is then the in vivo experiment with the rats, in which we investigated the effect of the red versus white meat, but also the effect of the fat content in the diet. So the first study, as I mentioned, was an in vitro study, in which we used the colonic microbiota, which we also call fecal inocula, of 5 healthy volunteers. And we added the colonic microbiota of these 5 healthy volunteers to 5 separate digestions of red meat. And as a model for red meat, we used beef. In the second part of this study, we selected 2 out of those 5 healthy fecal inocula and used them to investigate the effects of beef versus chicken digestion on the DNA adductome, and also see whether or not calcium had an effect in this. So these are the results of the first part of the study, in which we used the 5 different inocula. And you can see part of the heat map here on the right side of the slide. So what you can see is actually the difference between the DNA adducts that are present prior to the colonic digestion and the DNA adducts that are present post-colonic digestion. So to visualize this, what we did is actually subtract the pre-colonic levels from the post-colonic levels to see whether or not these adducts increased or decreased during digestion. So the ones that are marked in red are the adducts that increase, and the ones that are marked in blue are the ones that decrease during colonic digestion. Now, interestingly, the first thing you can notice is, of course, for all 5 experiments, we used the same meat sample, beef. And the first thing you can notice is that depending on the fecal inoculum that we used, depending on the colonic microbiota, we see different adducts being formed, different adducts decreasing over time. So there is really a high inter-individual variation in DNA adduction due to the colonic microbiota of these 5 healthy volunteers. Some rise, some decrease, and there's not really a clear trend which adducts do or don't during colonic fermentation. Now, of these 5, we selected 2 to perform the next part of this study, which is then actually compare what happens during the digestion of beef as a model for red meat and the digestion of chicken as a model for white meat. And then also, as I mentioned previously, we also supplemented the meat with calcium to see whether or not this had an effect. So again, here in this heat map, so you can see P1 is person 1, P2 is person 2. B stands for beef and C stands for chicken. And then CA added to that stands for the addition of calcium. So again, here in the heat map, you can see the difference between your, I hope I get a notion that my internet connection isn't stable. So I hope it's still coming through. So again, what we can see here, after colonic digestion and prior to colonic digestion, and what happens upon addition of calcium. One example specifically of interest to us is carboxymethylguanine. So carboxymethylguanine has been linked to red meat digestion previously. And as you can see, carboxymethylguanine, for example, rises or is higher during beef digestion compared to chicken, and also decreases in most experiments upon addition of calcium. So this one is definitely of interest to us because it is actively formed upon meat digestion of beef, but not chicken. And then calcium also exerts protective effects or potential protective effects. So both the meat type and calcium actually influence which types of DNA adducts that are formed and in which quantity they are formed. In the second study, so this was a follow-up study, we wanted to repeat the study on a larger scale, healthy donors, inocula. And furthermore, we wanted to assess whether or not additional myoglobin increased or decreased the levels of specific DNA adducts of interest. So in the first part, very similar to the first study, we could see that there was a lot of inter-individual variation. So depending on the fecal inoculum that was used, we could see that certain types of DNA adducts were formed or were not formed. And there were in total actually 26 different DNA adduct types that were associated with beef digestion specifically. Performing the multivariate statistics, we were also able to see that from those 26, there were actually 3 DNA adduct types that demonstrated significant potential as beef and are all alkylation adducts. So, which is of interest to us because of the heme hypothesis. So in the second part of this study, we wanted to see the effect of myoglobin. So we selected one of the 10 healthy volunteers to perform an additional experiments in which we performed digestion of beef and also additionally with or without myoglobin in 2 different levels. So we added 2 different concentrations of myoglobin to the beef to be able to compare the DNA adduct levels. And what we noticed was that indeed my adductome, some levels rose and some levels decreased. And in total, there were actually 34 DNA adduct types that went up as myoglobin concentration went up. And these are the 3 I'm referring to. So what you can see here is that these 3 DNA adduct types increase upon addition of myoglobin and are thus very much or could be very interesting and important in relation to the heme hypothesis and colorectal cancer risk in relation to red meat intake. In the third study, we performed the in vivo digestion with the Sprague Dawley rats. So as I mentioned, we gave them a red or white meat-based diet during 14 days, after which we sampled liver, duodenum, and colon. And these are the results in a heat map. It's part of the results because the table doesn't fit the slide, but it's just to show you as an example. So in the heat map, you can see the differences in DNA adduct levels between liver, duodenum, and colon samples, and then also depending on the diet. So in total, there were 4 different diets: a low-fat beef diet, a low-fat chicken diet, a high-fat beef diet, and a high-fat chicken diet. And as you can see, if you take a step back and you look at the heat map, the first thing I notice, so I hope you do as well, is that there is a clear difference between the tissue types. So looking at the heat map, you can see that the liver tissue stands out from the duodenum and the colon tissue. So the DNA adductome in liver is more different compared to the duodenum and colon, which are more similar, which absolutely makes sense, of course, because liver is an entirely different type of tissue compared to the bowel tissues. Secondly, apart from the prominent difference in tissue type, there was also a difference according to diet. So depending on the meat type and also the fat content, certain adducts increased or decreased. And in total, there were 22 DNA adducts that were of interest to us. So in general, of course, there's no time to really dig into the individual adduct types, but generally, we could see that there were several different types of DNA adducts that were formed during digestion, both in vitro and in vivo, and that meat type, calcium, myoglobin, and fat content all alter the DNA adduct content in the DNA. So in fact, we can very much expect that diet as a whole will significantly alter the DNA adductome. Now, as I mentioned, there were 4 studies in total, and this is actually a summarizing table of the red meat markers of those 4 studies. So what is listed here are the DNA adduct types that popped up multiple times in these independent studies. And can be listed as red meat digestion markers that may be of importance in the heme hypothesis. Now, where do they fit exactly in the heme hypothesis, or what does this mean this table, this list of DNA addicts? Well, as you recall from previously in the presentation, heme iron can induce the formation of N-nitrosyl compounds and the formation of lipid peroxidation products. And of the 7 listed DNA addicts, 4 are actually alkylation addicts, and N-nitrosyl compounds can induce alkylation of DNA. So they may fit in on the left side of this chart and be genotoxic or promutagenic due to N-nitrosyl compound formation. And then the other 3 are actually lipid peroxidation DNA addicts, which may be the result of lipid peroxidation due to red meat intake, which is of course then also important in the link with cancer initiation, promotion, and progression. Now in the future, we are planning to investigate this in vivo in humans. So we are setting up a colorectal cancer study with the Ghent University Hospital to study the formation or the presence of these DNA addicts in human patients. Now, over the years, we have also been working on the DNA Adaptomics methodology itself because DNA Adaptomics is in fact a very young technology and methodology. Actually, the first mention of Adaptomics or adductome was in 2006 in literature. And you may be aware or not, but in science, that is really not that long ago. So DNA Adaptomics is actually a really young field of research, and there still is room for optimization and further development of, for example, data processing workflows. So apart from implementing the methodology, we also invest time in further optimizing the methodology. And these are actually the results of one of our recent studies, which is currently in preparation for publication, but not published yet. So in DNA Adaptomics, we still have several things to tackle. So this is not a finite list, it's just a couple of examples. For example, in sample preparation, up until now, it wasn't really clear whether or not the enzymatic hydrolysis or the thermal acidic hydrolysis is the best choice to allow holistic or truly holistic mapping of the DNA adaptome. Also, regarding the analysis and the liquid chromatography separation, we envision the separation of a much wider range of DNA addicts. So even in our own method from 2015, we focused specifically on those alkylation and lipid peroxidation addicts, but there are many more to discover. So in this respect, we could tweak existing methods or try other methods. And in the data processing workflow, that really is the part where there is a lot of work to be done. Up until now, we have primarily taken over the workflow from metabolomics, but DNA Adaptomics, it's a whole world on its own. So it requires its own workflow. It requires its own optimization. For example, how to normalize, transform the data, and scale the data. So we've been looking into novel extraction, chromatographic, and pre-processing approaches for DNA Adaptomics. And in our recent study, we investigated 3 different aspects. One, the extraction, where we compared thermal acidic hydrolysis coupled to a solid phase extraction, compared this to enzymatic hydrolysis and fraction collection. Secondly, we assessed whether HILIC, so hydrophilic interaction liquid chromatography, could be of use or maybe even better compared to the reversed phase LC. And then also the third part, we assessed which type of normalization is best suited for DNA Adaptomics. So this is an overview of the setup we used to investigate this. So we again, we used in vivo samples to assure correct application. And we used tissue from, again, chicken versus beef-fed rats, of which we sampled liver, duodenum, and colon, which we pooled according to diet and tissue type to be able to re-aliquot them and compare them using the different methods. So in step 3, as you can see here in the slide, we compared the two different sample preparation procedures, as I mentioned previously. And then also in step 4, the HILIC versus reversed phase C18, to be able to interpret the data in step 5 and to see which method is actually better for DNA adaptome mapping. So the optimal normalization strategy was actually based on, or determination of the optimal strategy was based on QC clustering in PCA. I think the use of QCs has been explained, or I hope it has been explained in the previous lectures. So normalization was assessed based on QC clustering and also the total number of valid OPLSDA models that could be constructed after normalization. And what we noticed was when comparing QC normalization to internal QC normalization to loose, that we could see that QC normalization was in fact the best option. So these 3 types of normalization are commonly used in metabolomics, but apparently QC normalization is also the better option for DNA adaptomics. Now, this is an example, and it's quite small, so I'm not sure whether you will be able to see the details. But the overall picture here is, in fact, on the left, you have the normalization strategies using LOS or loose, using different scan smoothing parameters. And on the right, you have the IQC, and bottom right, the QC. Now, as you can see, the QC is marked in green because it shows the best clustering of QC samples, and also what is not shown here in combination with the most validated OPLSDA models. And these are the results of 1 out of 4 data sets, which was in fact the thermal acidic HILIC data set. But for the other 3 data sets, results were very similar. And also there we could see that QC normalization was the better option, apart from 1 out of 4 where IQC was a better option. But overall, I would prefer to work with QC normalization. Regarding the optimal extraction and LC strategy, as I mentioned, we evaluated thermal acidic versus enzymatic hydrolysis, and also the use of HILIC versus reversed phase LC. And here we based the results on the total number of valid models and their validation parameters. So the models that we're interested in is specifically 3 tissue discriminating models to model the difference between the 3 tissues, and also 3 diet discriminating models to model the difference between the 2 diets in the respective tissues. So from previous work, we know that there are differences according to tissue and according to diet. So our DNA adaptomics methodology should be able to map these differences. So depending on which out of 4 methods can actually document these differences and provide us with valid models is the better option for us. So this is a summarizing overview of those model parameters. And as you can see, the thermal acidic HILIC data set has 2 valid models and 2 borderline valid models, whilst the other 3 merely have 2 valid models, or even less than that, only 1. So all in all, the TA-SPE HILIC method appears to be the best method for DNA adaptome mapping. And with DNA adaptome mapping, I'm specifically referring to mapping of all ions present in the sample without really digging into the data set. So this is merely looking at the modeling potential. So again, I will not go into details on the study, and soon there will be a publication if you're interested to read up on the details. But generally, we could see, or it was our conclusion, that regarding sample preparation, the thermal acidic hydrolysis coupled with solid phase extraction and HILIC is a very good starting point for further optimization and exploration of DNA adaptome mapping. So comparing thermal acidic hydrolysis and detection of nucleobases versus the enzymatic, and detection of nucleosides, the nucleobase-based approach appears to be the best one. And for normalization, as I already mentioned, QC normalization is preferred over IQC or loose normalization. Now, this is, of course, very detailed regarding the DNA adaptomics. But what is important to remember from my talk today, or what I would like you to take home from my talk today, is in fact the what, why, and how of DNA adaptomics. So what is DNA adaptomics? DNA adaptomics is measuring DNA adducts as markers of exposure, biologically effective dose, and effect, which is, of course, highly useful information for exposomic studies, but also dietary interventions, etc., which is the why. And then last but not least, the how. So when wanting to perform DNA adaptomics, it's best to use high-resolution mass spectrometry to enable both targeted and untargeted analysis using a fit-for-purpose workflow. So without further ado, I would like to thank you and end my talk. Thank you all for joining today and for your attention, and most definitely also to my team and more specifically Professor Lien van Haken for all her support throughout the years, and also for our most recent work in collaboration with Professor Silvia Balbo and her team. Also a big thank you to them.
Right. Thank you, Liz, a lot for that interesting talk. Okay, so perhaps we can proceed with the Q&A portion at this point. Brian, if you may allow me to read the first question. This is from Gert.
Sure.
I think it's Gert or Gert. Anyway, the question is, do you think DNA adaptomics would be a useful technique to explore effects in environmental pollution on humans? For example, I think it would be interesting to see if DNA adducts on PBMCs can provide a marker of exogenous toxins exposures.
Yes, most definitely. For example, one of the projects we have recently started is an exposomics project here in Belgium, where we want to assess the exposure to POPs, so persistent organic pollutants, and see whether or not this indeed results in DNA adduct formation. So yes, most definitely. There is, I must note that there is still a lot unknown about DNA adduct formation, but I do believe that it's very useful to study it in humans because it will learn us more about exposure. Some pollutants aren't stable, so it might be unclear whether or not someone was exposed or not. So DNA adducts can help in that. And then also, of course, relating to potential adverse health effects, it's definitely something that I and many others are interested in looking into. Yep.
Right. I have a question actually, because I'm a bit interested in exposomes as well. In recent years, I had
damage.
Yes, I had the training for micronuclei formation. So essentially, it's quite related to, I don't know, it's quite related, but definitely I'm looking into exposomes. My first question is that you informed us earlier about the importance of having a good DNA yield before pushing through all this experiment. So do you think there's a specific threshold where we can actually say that this yield is good enough to push through with the experimentation?
Yeah, I would say that 50 micrograms is the minimum, but I would definitely prefer to go more towards 100 micrograms, if possible. It depends on, of course, it depends on the type of study and the type of tissue and the availability of the tissue. But 50 micrograms is the absolute minimum for DNA adaptomics. Yeah.
All right, thank you for that. Also, another question is that looking through the internet, I think most of the kits that are available commercially for DNA adaptome studies, particularly on extracting the DNA available on a specific sample matrix, the available ones are for immunoassay or ELISA works. The ones that you use in your laboratory are this in-house formulation of DNA extraction?
No, for DNA extraction, we use also commercially available kits. So I know there are several labs who have their in-house methods for DNA extraction, but we most often use Qiagen kits or Magrinago. I'm not sure how to explain it actually. It's a German.
I think it's Mashere. We used to pronounce it as Mashere here in the Philippines.
Yeah, but they are available, and those are the kits that we use as well. Then depending on the tissue type or the sample type, we use the different sets.
All right. Before we proceed with the question coming from YouTube, last question coming from me. When is the best time to set your research design in like, for example, are there indications on when to use or target nucleotides, nucleosides and nucleobases for that matter? So when would be the best time to choose among these three options?
Well, first of all, during method development. So you, depending on your setup and your study, you can do a literature search and see which type of adducts that are already known in your specific area. And depending on that, you may look into the methods that already exist and that already work. I think most of the methods will be nucleoside-based methods, but one of the downsides to those methods is that the sample preparation is quite extensive. And if you want to perform high throughput analysis, it's not as feasible to do it as such. And it also depends on the type of adducts. So most of the adducts are indeed formed at the N7 and N3 position of guanine and adenine. And if they're that type of adducts, you can use whatever method you want to use. But let's say that you have a compound which forms adduct specifically at the phosphate group, you need to go for a nucleotide method.
Thank you for that, Liz, a lot. We have a question from YouTube. This is from our former colleague, Dr. Sofia Amara. In order to identify DNA adducts, do we also need to refer to a mass spectra database? She's actually asking if there's such a thing as a mass spectra database.
It's a good question because not myself, but colleagues in the US are currently working on a mass spec database for DNA adducts. For DNA adducts, there isn't an actual database, not just naming the adducts, but most definitely not with the spectra, but it is a work in progress. Sometimes you can find spectra of adducts in other databases, like for example, the human metabolome database. Some adducts are included there as well, but there's no, to date, there's no existing dedicated DNA adduct database. It's under the way, yeah.
Perhaps in the future, be able to go back to populate a database on DNA adducts. Brian, anything you want to add?
I have questions myself.
All right.
On a biological, first of all, thank you very much, Lotz, for such a wonderful presentation. When I first had a discussion with Jay, because there's a big interest on DNA damage at FNRI. So I was quite excited when I learned that you would be available to give us a talk, because it rounds everything up, and the actually what you presented is really a future advancement in the field. So it's good that you were our last presenter, because it shows you, it combines the expertise already available at FNRI for genetic studies, and also the future interest within in the Philippines for mass spec-based analysis. So thank you very much. On a biological perspective, I'm just curious, because DNA adducts, especially if you have a methyl adduct, it's very well studied, right? And methylation, it has very big effects. I'm just curious if the effect of a certain, let's say, malonaldehyde adduct would impart the same biological function as a methylation on that specific DNA, and how long does that adduct stay in the DNA? Is it a permanent adduct or not?
Yeah. So the effect is not the same. So depending on the adduct type, it could be mutagenic or not. For example, you have N7-methylguanine and O6-methylguanine. So N7-methylguanine is formed more abundantly than the O6-methylguanine, but O6-methylguanine is more harmful. So there is a difference, and even if it's a small difference regarding the potential effects, it's important to make a distinction between the different types of adducts and study the effect per adduct. So you cannot take the whole of all adducts and see where that leads. No, it's really a matter of the individual adduct and its effects. And apart from the adduct, the position, of course, where it is adducted is also of importance. But unfortunately, this is something that is vastly understudied. And for some DNA adducts, it is known that they can, for example, induce transversions and are linked to, for example, mutations in P53. But for some, nothing is known, and it's still under investigation or should be investigated in the future.
Okay, thank you. Yes, sorry.
Yeah, regarding the stability, it's a bit the same. So some adducts are easily repaired by the existing DNA repair mechanisms, and some are not. So for certain adducts, you can find this specific information in literature where they've established that it is repairable. And for some, they have not been able to find a working repair mechanism, and I see that it accumulates over time. And also this is adduct specific.
Okay, so still in keeping with the biological effect and methylation and duration. In fact, I am curious as well. So you mentioned, you did give a wonderful presentation about the use of adaptomics as, let's say, a food intake biomarker for red meat. And as we know, red meat intake is associated, of course, it's still, in fact, it's still being debated right now, the body of evidence, but of course, there is an understanding, of course, unhealthy diets lead to poor cardiovascular outcomes. I'm just curious if there is really a study available linking directly DNA adducts and cardiovascular diseases, for example. If there's already strong epidemiological evidence for that.
No, there are studies linking red meat intake to cardiovascular diseases, but not DNA adducts specifically. So DNA adducts up until now have primarily been investigated in relation to cancer, because that, of course, makes sense. You have DNA damage, mutations, cancer. But in relation to other diseases, I'm pretty sure there are only a handful, maybe, or maybe none, studying that. I think regarding the developmental aberrations, the paper I had in mind was in fact a study that investigated DNA adduct levels in wildlife, in embryos and newborn animals, and they could link that to developmental aberrations. But it's one of very few studies focusing on that. So there's a huge gap there.
That leads me to my pitch. In fact, we do have a birth cohort. So it would be quite, Jacobs, let's think about this. It would be quite interesting to see if in the cord blood of children, there would be some differential adducts, which would lead to us to, get insight into the exposure of the mother during pregnancy. Yeah. And then we can link that to growth outcomes of the child. Okay,
True.
let's have a discussion on that. Listen up. That would be quite interesting.
It's an interesting twist, I guess.
Yeah. But just last question on a chemistry perspective. I'm curious. You said so. What was it? Chemical. TA. What does TA mean? I just wrote on my note. TA SPA helic. Thermal acidic. Yeah. I'm curious about the SPE that you used. What kind of SPE is this?
It's just I don't recall the name. It's just a regular C18 SPE.
Yeah, so that brings me my curiosity. Isn't it ironic though? So you first do an SPE on a C18, And then you said that the ones that are stuck to a C18 SPE column are better separated on a helic than a C18 itself. I mean, that's quite opposite, I think.
Yeah, it's the thing is with the SPE columns, it's the method that we use in combination with our own C18 RPLC method. So it's an established method, and that's why we wanted to try it. And then indeed, we mix up the different methods, and then in the end, we get the helic giving the best results. But what's behind it is actually something that requires further investigation, because of course to us as well, it's not clear why that is exactly. I do know that, for example, the helic is better suited for the separation of more polar adducts. And in our specific setup, we're looking into intertissue differences and also dietary induced differences, which primarily, so the diet we expected to primarily induce differences in the alkylation and the lipid peroxidation adducts, which are quite polar, specifically the alkylation adducts. And then in the tissue, we mostly expect endogenous DNA adduct types to be different between the two tissues, which are also more polar.
Okay.
So that respects we concluded that helic may be indeed better in this specific setup to be able to separate those polar adducts. But those polar adducts, they also come off the C18 SPE column. So
yeah, well, it's just a quite interesting, chemically speaking, it's a very interesting combination. But yeah, thank you very much. Do we have more questions, Jacobs?
Yeah, I think we have to entertain this last question from the Philippine Genome Center Mindanao from Sir Michael Bacchus. He is asking if there are efforts to develop methods to investigate RNA adducts, or can we use the existing DNA adaptomics methods to, can they be modified and used to specifically investigate the RNA adducts?
So you can indeed, specifically the nucleobase methods, you can use for RNA adaptomics as well. So you can just, the only difference is that you will have a different sample preparation. You will isolate RNA instead of DNA. But after that, I think the method can be transferred and used as such. Yeah.
All right. Thank you for that, Lizella. So this has been an interesting discussion. I'm sorry, Brian, but I have my own bias with genomics. It led me back to home. So thank you, Dr. Hemerick, for that very informative discussion this afternoon. So mga kababayan, isa pong palakpakan para po sa ating speaker. A virtual round of applause to our speaker, Dr. Lizelot Hemerick of the Ghent University. Thank you.
Thank you very much, Lizelot. Thank you.
Thank you for having me.
Thank you. Thank you. So actually, we will not let our 9-day engagement to end without a simple closing toast. So we're now moving into our short closing program. And the organizers actually thought of giving the virtual floor to our dear participants at this point. So early this week, we actually sent emails to some of our participants who we thought are very active in engaging with our speakers. Our objective of sending them this email is that we wanted to hear from them, their feedback and insights about the 9-day OMICSena lecture series. Although, of course, this might not be the perspective of everyone, we hope that we were able to provide you with the needed information about the other omics platforms on nutrition and health research. So actually, early this morning, we're about to give up kasi walang nagpapadala ng email. But this group of participants from the University of Santo Tomas actually sent us a video message, and here's what they have to say. So maybe we can roll the VTR now.
Thank you for the kind invitation dito sa siyam na araw ng OMICSenang pag-aaral at pagtuklas. In this program, we all came to realize the impact of omics in expanding our search for knowledge and truth to the molecular level. I personally see it as our springboard for opening our creative minds to find answers to many questions that will address our nation's needs in nutrition and personal health. We are very lucky to witness the initiatives of the DOST-FNRI to bring scientists and experts all over the world from all relevant disciplines and humbly present their brilliant research practices, available resources, and tools in the fields of metabolomics, lipidomics, bioinformatics, and collectively, I must say, this is indeed nutrigenomics. Your great presentations steered our imagination, drives our passion, and open doors for collaboration. The truth is, we found some good collaborators in this program. And as we speak, my teammates and I are hoping and praying that our humble attempt to collaborate in your labs will soon materialize. I am moved every time I hear we cannot do omics alone. We need the expertise, resources, and experiences of each other so that in the end, the society in general will benefit from our collective works. And lastly, I dream that we move forward in our quest to support our omics community needs by perhaps starting our own nutrigenomic society in the Philippines. It's only a dream, but what is real now are the experts. Thank you to our Balik scientists, Dr. Gerard Brian Gonzalez, the esteemed panel of speakers, our great host, and DOST FNRI team under the strong leadership of Director Dr. Imelda Angeles Agdepa. Together with my teammates, we say,
Thank you, DOST FNRI, for hosting OMICSena.
magandang araw po sa inyong lahat.
Hey, maraming salamat po, Ma'am Marites Catyon and the team for your feedback, and for the rest of our participants who joined us from day one up until now, and to those who joined us in the middle of the series. Sa man po kayo nagsimula para masubaybayan kami, maraming salamat po sa inyo. We are actually joined now by our former director, Dr. Mario B. Capansana. Hello sir, magandang hapon po. So I will now give the floor to the Supervising Science Research Specialist of the Nutritional Biochemistry Section of the Nutrition and Food R&D Division, Mr. Michael Sirafico, to give us a walkthrough on what transpired and the entire run of the OMICSena lecture series. Over to you, Sir Kim. Magandang hapon po.
Magandang hapon, Jay. Thank you. Am I being heard?
Yes, sir. Loud and clear po.
Okay, salamat. So good afternoon once again. My task for today is to bring you back to what has been a successful 9-day running of the OMICSena lecture series. Whether your participation was a targeted activity for the past 2 weeks or it was untargeted, I wish to thank you for staying with us from day one till today. OMICSena emanated from one of the 3 important pillars of the DOST-FNRI to disseminate what we do and how these things that we do bring forth knowledge, collaboration, action, and service. The first season of OMICSena is a 4-hour talk on the frequently asked questions about nutritional genomics. Last March, the unit head of the Nutritional Genomics Unit laid out the fundamentals of nutritional genomics. From there, advocacy started. Now on its second season, we binged to 9-day omics field episodes. Behind the scenes, it took the DOST-FNRI team 2 months for the pre-production process. But thanks to our DOST Balik scientist for metabolomics, Dr. Gerard Brian Gonzalez, for bringing in his esteemed colleagues, OMICSena lecture series came to life. The OMICSena lecture series kicked off with a 20-minute virtual launch that was graced by the DOST Secretary, Honorable Fortunato T. de la Peña, DOST Undersecretary for Research and Development, Dr. Rowena Cristina L. Guevara, and DOST FNRI Director, Dr. Imelda Angeles Agdepa. It was followed promptly by the first lecture on the Introduction to Nutritional Metabolomics by Dr. Gerard Brian Gonzalez. He imparted the important space occupied by metabolomics in the omics pathway and how the study of the metabolites could pave the way to understanding the mechanisms of the established nutrition biomarkers and shed light on the discovery of potential new nutrition and health biomarkers. During the first lecture, Dr. Gonzalez provided a preview of the 9-day lecture series. Day 2 was all about the concepts, definitions, and classifications of biomarkers. Dr. Lars Ove Dragsted shared his expertise with us, particularly in finding biomarkers of food intake with the use of metabolomics. Dr. Dragsted held that the accurate classification, validation, and combination of food biomarkers could lead to an enhanced classification of recent intakes of foods and drinks and will help in identifying potential misreporting in short-term dietary assessment. Day 3 was a back-to-back discussion on the metabolic profiling for biomarkers of enteric infections and malnutrition and mass spectrometry-based analysis of the metabolome by Dr. Gordana Panic of the University of Southampton and Dr. Gerard Brian Gonzalez. Dr. Panic shared her real-life experiences about the utility of metabolomics and metagenomics in randomized controlled trials. For his part, Dr. Gonzalez comprehensively shared his expertise in metabolomics workflow with mass spectrometry as a platform. On day 4, we listened to Dr. Albert Kuhlman of the University of Cambridge. Dr. Kuhlman had 4 key takeaways from his talk. First, there is no perfect method to measure everything. Second, each research design requires the most suitable method. Third, new insights can be gained when we integrate new approaches in data analysis. And fourth, we need to keep in mind that experimental designs and methods have their own limitations. Also on day 4, Dr. Gonzalez provided a hands-on experience and simplistic simulation of metabolomics data analysis using free online software. As we take it from here, we are looking forward to working on real local data such that in the future, we can gather again to experience another hands-on simulation of metabolomics data. For day 5, OMICSena flew us to Parma, Italy to have a glimpse of food omics. Dr. Laura Righetti of the University of Parma shared with us a case study in plant food metabolomics. Here we get to know that food omics can be applied to study the safety, quality, authenticity, and bioactivity of foods. Like the rest of the other speakers, Dr. Righetti emphasized the importance of collaboration to achieve the goals of the multi-omics approach in food and nutrition research. The topic for day 6 is quite advanced, but thanks to the detailed and comprehensive presentation of Dr. Justin van der Hoof of the Wageningen University, we get to retain important concepts in computational bioinformatics. It is quite surprising that through an interactive discussion, Dr. van der Hoof has a following in our midst. The OMICSena team is looking forward to contributing to the next generation of bioinformaticians through this series. On the 7th day of OMICSena, Professor Jonathan Swan tackled one of the relevant research agendas in the country today, early life malnutrition, particularly stunting. Dr. Swan introduced nuclear magnetic resonance, or NMR spectroscopy, and how the platform can be used to study early life health and nutrition disruptions. He underscored the current applications of NMR-based metabolomics in understanding undernutrition, microbiome, niacin nutrition, and biochemical aging among children. Yesterday, on our 8th day, Dr. Lynn Van Hacke left us all in awe as she shared with us her research passion. What we learned from her talk is the immense possibility of generating tangible systems inspired by R&D work. Who can forget her proposition for point-of-care metabolomics testing and her advocacy to bring physiology and biochemistry on the table after being momentarily left out by the sudden influx of genomics-based research? And earlier, Dr. Liselot Hemmerich introduced us to the concept of DNA adaptomics. I believe her talk is still fresh in your minds, so I would like to reiterate her take-home messages on what DNA products are, and they are the markers of exposure, biologically active, effective, and those in effect. Why do we have to study DNA adducts? Because it provides highly useful information, such as in exposome studies and nutrition studies. How do we measure DNA adducts? Through high-resolution mass spectrometry to enable both targeted and untargeted analysis, and through using of a fit-purpose workflow. We owe the success of this 9-day lecture series to you, our audience. The concept of OMICSena is to create good scenes to impart learnings that the Institute believes to be relevant not only for its in-house researchers, but also to you, our stakeholders and partners. DOST-FNRI is also in immense gratitude to Dr. Gerard Brian Gonzalez for helping us plot this series. As they say, there is power in synergy. Thank you for being a catalyst, Dr. Gonzalez. The series tallied an average of 280 daily Zoom participants, and we are overwhelmed by the turnout of registrants that spans other Asian countries, including Australia and the Middle East. We expect that OMICSena will contribute to expanding our streaming footprint on YouTube, as you can stream the entire series on demand, all you want. As today is our so-called season finale for 2021, we hope to see more of you in the next season of OMICSena. Help us enhance this advocacy of the DOST-FNRI by letting us know what you want to see next in the omics sciences. We are closing the curtains now, but rest assured that we will come back. Till the next straw. Keep safe, everyone.
Maraming salamat po sa synthesis. Sir Kim, thank you for the synthesis. So, in any performance, it is actually customary that we have what they call the curtain call. So if this isn't too much to ask, please spare me a minute of your time to present to you the men and women behind the 9 days of the OMICSena lecture series. It was actually me, Diane, and Frances, whom you actually see on your screens every day. But these guys were actually the ones who sent you the registration link. They created and posted the art cards in our social media pages, sent you the Zoom links, and continuously, up until now, flooding our chat box with the attendance and evaluation links. Together with the support of the DOST-FNRI management, led by Dr. Imelda Angeles Agdepa and our Deputy Director, Dr. Anthony Calibo, the OMICSena platform is actually a partnership between the Nutrition and Food R&D Division and the Technology Diffusion and S&T Services Division, led by Engineer Rosemary Garcia and Dr. Miltor Gonzalez. Guys, please open your cameras so our audience can actually see us for our curtain call together. Okay? Ayan po sila. Sila po, yan po si Ma'am Marie, si Dr. Calibo, and the guys. So actually are sending you the emails and the links for your registration as well as the evaluation and attendance. So mula po sa aming lahat dito sa DOST-FNRI na naghatid po sa inyo ng OMICSena lecture series, maraming salamat po sa pagsama sa amin sa loob ng 9 na araw. Thank you po. Maraming salamat. So, ayan, maraming salamat po sa inyo. Thank you guys. Thank you. So, actually, mounting a stellar lineup of speakers would not be possible without the effort of this man, whom the DOST-FNRI is fortunate enough to stumble upon last April 2020. He is actually the second balik scientist that DOST-FNRI has hosted, and the Institute is grateful to have Dr. Brian Gonzalez, who worked with us and guided us as we initiate the OMICSena lecture series. Dr. Brian Gonzalez, I'm now giving you the floor for your maybe insights or views before we end the OMICSena lecture series.
Well, I just want to thank FNRI, of course, for hosting me. It's a pleasure and an honor for me to be part of this initiative, and especially to Jacobs, who I have been constantly in brainstorming sessions with. So thank you very much. It is my hope that this initiative would help build the capacity in the Philippines for metabolomics research. And I hope this was just a start, and it stirred the interest and curiosity of different labs in the Philippines for this. And of course, with this 9 days webinar series, it's my hope that at least some of you learned something new and are willing to collaborate indeed for us to push this type of technology forward in the Philippines. So yeah, thank you.
Maraming salamat, Doc Brian. Maraming salamat for bringing us your peers and of course your expertise. No, you actually given us 3 lectures, and we're looking forward for more. Pwede pa ba kaming humirit for the next round of the OMICSena lecture series? Okay, thank you, Doc Brian. And to officially bring down the curtain for this season of the OMICSena lecture series, may I call on our dear Deputy Director, Dr. Anthony P. Calibo, for the closing remarks. Over to you, Doc Anthony.
Thank you very much, Sir J. Costancis, Dr. Brian Gonzalez. Of course, I'd like to give some salutations to our DOST Secretary Boy de la Peña, Science and Technology Undersecretary for Research and Development, Dr. Yusek Guevara, the DOST FNRI Director and Scientist too, Dr. Imelda Angeles Antepa, our speaker for this afternoon, Dr. Liselot Hemrick of the Ghent University, the Nutrition and Food R&D Division Chief, Engineer Rosemary Garcia, likewise to the Division Chief of the Technology Diffusion and Science and Technology Services Division, Dr. Milford Gonzalez, the Nutritional Genomics team, other esteemed research specialists of the DOST FNRI and other DOST attached agencies, sectoral councils and collegial bodies, colleagues, virtual guests from other government agencies, various professions and disciplines, the academe and higher education institutions, omics enthusiasts, ladies and gentlemen, good afternoon, good morning, and good evening. We have reached the end of the virtual OMICSena lecture series held on Zoom and live streamed on YouTube, but this marks a new beginning of learning and more opportunities for the advancement in omics technologies. I would like to express my sincerest gratitude to our speakers from the European Union and the United Kingdom, and likewise to our DOST Balik scientist, Dr. Gerald Brian Gonzalez, for working with us in this endeavor. Likewise, the presence of our former director, Dr. Mario Capansana, who was also instrumental in conceptualizing and the inception of the Nutrigenomics Laboratory for our institute. Our heartfelt thanks to all of you for imparting your knowledge, time, and expertise. The great Benjamin Franklin, a renowned innovator and personality in science and technology, once said, "An investment in knowledge pays the best interest." To all the participants who opted to spend 9 learning field afternoons with us, my deepest gratitude goes to every one of you. I think this is a version of a 9-day prayer, but it's a prayer to advance omics sciences and understanding to benefit the food and nutrition research for our country. It is because of all of your active participation that this webinar is a success. All of you are our inspiration to share more information and to innovate further despite the challenges brought about by the COVID-19 pandemic. Thank you, Sir Kim, for your synthesis. I appreciate the 9-day review, as this has also given me a glimpse of what has taken place, as my busy schedule also prevented me from logging in all of those days of the OMICSena. But I'm very sure that every day that this has been conducted was always a resounding success. To all the working committee members behind this webinar, especially the nutrigenomics team, the supports team coming in from the Technology Diffusion, Science and Technology Services Division, of course, the use of the mini theater, and likewise the IT support team of the DOST-FNRI. A massive thanks for your continuous hard work in delivering learning and development platforms that can change the way we live today and the way our future generation can enjoy the perks of life. Thank you, Dr. Agdepa, Engineer Garcia, and Dr. Gonzalez for guidance and unwavering support to this endeavor to your research specialists. This webinar could not have been smooth running without all of your commitments to food and nutrition research. Magandang hapon, maraming salamat, mabuhay po tayong lahat.
Maraming salamat po, Dr. Calibo. Thank you for voicing out our aspirations and our hopes for joint efforts linked by the OMICS platforms. Thank you po, Doc. So, total naman po huling araw na po ito ng ating series, no? Baka naman po makikisuyo kami. Buksan po natin yung ating camera para po sa ating photo ops. It has been a tradition here at the Institute that we actually have this photo ops after each of the webinar series. So, bukas po tayo for the photo ops. All right. Someone from the technical committee would be doing this. We'll do the screenshot.
Okay po.
Okay.
First panel. 1, 2, 3.
Okay.
Second panel. 1, 2, 3. Third panel. 1, 2, 3. Fourth panel. 1, 2, 3. Fifth panel. Last. 1, 2, 3. That's all.
Okay, so maraming salamat po sa inyo. This has been the OMICSena lecture series, and please stay tuned for more. But for now, goodbye and thank you for joining us. Ingat po tayong lahat. Salamat po. Thank you, Dr. Calibo. Thank you, Ma'am Marie.
Congratulations to the OMICSena team. Well done.
Thank you all. Thank you.
Thank you, Dr. Calibo.
Thank you po.
Thank you, Doc Brian.
Dr. Mark, thank you for joining.
Thank you, Dr. Mark.
Kumusta na kayo? Very good.
Salamat po.
Salamat po. Salamat po.
Jacus, thank you for the MC.
Salamat po. No, you're welcome po, Doc. No problem po.